Hello, it is noon. Thank you very much for joining. Today, our seminar speaker is Dr. Katie Howe, who is currently a postdoc here at Dauphin Island Sea Lab, and she's working with Dr. Brandy Kiel, Reese, and myself. The title of her talk is My Journey as a Microbial Oceanographer and an Introduction to Silica Cycling in the Arctic. Dr. Howe comes to us from doing a PhD at Florida State University. She did a master's at Nova Southeastern, and she did her bachelor's at the University of Tampa. Currently, she's got two of her dissertation chapters published. She's got one of her master's, a master's publication as well, and she's being productive there. Some other fun things about her is, as you're going to see, she's done largely kind of a PhD at the computer. But despite having so much time in front of a screen, she is working towards her and nearly there, her 100 days at sea, which is pretty awesome. And when I see at sea, I'm not meaning like go out on a boat for an hour and then come back in. These are like full days at sea on big research vessels. So that's pretty cool. She's also what I like to call an Arctic tern. She has done the transpolar route in less than a calendar year, which was pretty amazing. So she, as she's going to discuss, she's been from Svalbard in the Arctic down into the Ross Sea. And beyond that, she's been active in outreach and some other information that I think is going to be interesting. She is moonlit at a brewery. So I'll leave that there. But I'd like you guys to welcome Dr. Katie Howe.
Hello, everyone. Thank you, Jeff, for that introduction. So I wanted to tell you a lot about my background since I have been here for a year and a half, but I haven't really interacted with all of you yet. So to start at the very beginning, there we go. I'm originally from Massachusetts. I love to learn about all things, especially science-related things, and I am extremely motivated by travel. So my first international trip was when I was 12 years old. I spent 9 weeks with my aunt, uncle, and cousins in Thailand. And I just want to give a quick shout out to my uncle, who's also an oceanographer and engineer. His name's Bruce Howe. He currently works at the University of Hawaii, and his passion project for the past few decades has been smart cables, which are science monitoring and reliable telecommunications cables. So essentially, they're these contraptions on the trans-ocean telecommunication cables that have different sensors on them. So bottom pressure, temperature, and seismic accelerator are a few of the most important ones. And this has real-time applications. These sensors are connected to satellites, and they're very important in terms of, for example, earthquake and tsunami monitoring and warning systems. My uncle was a big influence in terms of my scientific career growing up. I never know where he was going to travel to next, and he actually has over 550 days at sea. A few years later, I got certified to scuba dive, and then the next year I did a shark research program in Fiji, where we spent a month diving with bull sharks, and we each did a little independent project on it for college credit. So going into college, I definitely thought I was going to be a shark scientist or a nature photographer at the very least. So I went to the University of Tampa and got my bachelor's in marine science and biology, and I was able to take some really interesting courses there. So the first one I did was called Chemistry and Art, and that taught us about art restoration, different materials used for art, as well as the biogeochemistry of glass blowing, for example. I did a semester in Broad studying sustainability in Perth, Western Australia. And then I did an elasmobranch biology summer course at the University of Southern Mississippi. And for those of you who know Jeff's previous lab manager, Lindsay Lemke, that's actually where I met her for the first time. I took a few years off after undergrad. I didn't feel like I had the confidence to pursue a graduate degree right away. But actually, 11 years ago today, I applied to Nova Southeastern University for their Coastal Zone Management program. I worked with Dr. Alexander Slolyev in his physical oceanography lab. I didn't have the lab experience to get into the shark biology lab, which is what I really wanted to do. But thankfully, this project allowed me to actually do my lab work in the shark biology lab. So I got to still work with those people. So my thesis was on surfactant-associated bacteria in the sea surface microlayer, which is essentially the air-sea interface. But then I worked on a few side quests as well, including oil droplet propagation, models, and lab experiments. I also did a lot of data collection for a study on hurricane trajectory and rapid intensification in the open ocean. So these photos on the right, 3 of them were from my main research cruise during my master's. And so that was called the laser experiment. This was done by the Gulf of Mexico Research Initiative. And the main point of this cruise was to release 1,000 drifters and 10,000 drift cards and follow their trajectories in the Gulf of Mexico so we could study those small scale currents. I was one of 2 women on this ship and the only biologist, so that definitely made for a very interesting experience. And then another part of my research was working with satellite images. So the photo in the bottom right corner is a picture of me in my lab at the German Aerospace Center in Neustrelitz, Germany. The month before I defended my master's, my advisor thought it would be a great idea to go frolic around Germany and go to 3 different locations of the German Aerospace Center and let them know what we've been doing with the images they've been providing for us. And as Jeff mentioned, this was published, thanks a lot in part to Dr. Kayla Whitney Dean, who was a PhD student in the same lab at the time. So my PhD was in biological oceanography at Florida State University. I worked with Dr. Olivia Mason. The picture on the top left is just to tell you once again that this PhD was all computer coding and writing. There was a new PhD student coming in when I was getting ready to leave who did not feel confident doing fieldwork internationally for the first time. And so I happily took that on and was able to go to Africa for 3 weeks and in Namibia. And that was actually the first time I've ever seen a sediment core in person. So my dissertation topic was identifying novel hydrocarbon degrading microbes in the ocean. So one of the most common questions I got during my PhD, besides when are you going to graduate, was do you grow these bacteria in the lab? And when you realize that only about 1% of bacteria can be grown in the lab, you realize that the coding that I do is very important. So to put this in a visual perspective, here's a tree of life from Hug et al. 2016. And I just want you to notice all the red dots that are on it. Those represent groups that have not been able to be cultured in a lab. And so how do we find out about these microbes? We do shotgun metagenomic and metatranscriptomic sequencing. So that's when you sequence all of the DNA and RNA in a sample. So throughout this talk, metagenomic will be abbreviated MG. And I want you to remember that's all the DNA in a sample, and it tells you about who is there. Metatranscriptomic will be abbreviated MT, and that's all of the RNA in a sample, and that will tell you about activity. So here's the schematic I really like from Yang et al. 2021 that shows the different parts of microbial sequencing. So in the left-hand panel under A, there are 2 biological sample collection methods. The first one is culture-dependent work, and that's when you actually grow these microbes in the lab. DNA extraction and sequencing, of course, and then quality control and other coding. That's mostly straightforward because you know exactly, well, you should know exactly what's in the cultures, so you know what to look for. I did not do any of this for my PhD, so I'm not going to get into many details of that. What I did do was the culture independent work. So for my PhD, all of my samples were already collected for me and sequenced. And what that entails is DNA extraction, sequencing that DNA and RNA to get the clean, quality controlled reads. And then moving over to panel B, this is what the computer coding is all about. So there are two different ways to go about this. The first is assembly-based approach, and this is where we obtain metagenome assembled genomes, or MAGs. So these are sequences that represent a microbial genome. We can then go ahead and do gene prediction and annotation, so find out where those genes are located and what they do. And then we can also do taxonomic profiling to look at phylogenetic relationships. There's also a reference-based approach where we use publicly available genomes and we see how those compare to our metagenomic and metatranscriptomic data. And we do what's called read mapping, where we take the sequence of the gene or genome of interest and compare it to our DNA and RNA reads, and that will give us a count number, and that will tell us about abundance and activity. So essentially, my workflow is broken down into three main steps. We obtain the metagenome assembled genomes, or MAGs. We search for their close relatives using whole genome phylogeny. And then we do read mapping to determine abundance and activity. So this is the second chapter of my dissertation, and it was published last year on novel, active, and uncultured hydrocarbon degrading microbes in the ocean. And so the samples I'll be talking to you about for this were taken during the Deepwater Horizon oil spill. Before I go into the environmental impacts, I think it's always worth noting that this was a huge disaster where 11 people were killed and 17 were injured. And the spill lasted for 87 days. It was from April to July in 2010. Over about 4 million barrels of oil were released. And this spill was unprecedented in terms of its depth and the volume of oil released. So on the left side, you can see a map of the Gulf of Mexico, where the rig was and the extent of the oil spill. And then on the right, you'll see a schematic of what happened to the oil when it was released. So the oil that reached the surface of the ocean was photooxidized, evaporated, burned, maybe even skimmed. But you can see that there's a plume of oil at the bottom. And the main method of removal here was by microbes. So microbes would come in and eat the oil in this deep sea plume. So the Macondo oil, which was MC252, was a complex mix of hydrocarbons, as are many types of oil. Mostly it was made up of saturated hydrocarbons. They totaled about 76%, and aromatic hydrocarbons were also present. And those are the two groups that I'll be focusing for this section. So why did we do this? Initially, with these Deepwater Horizon studies, there wasn't a clear link between taxonomy and function. There were a lot of papers that would talk about taxonomy and what bacteria or other microbes were there. And then there were separate papers that would talk about what genes were being expressed. But now that we have updated bioinformatics tools and databases, we are able to find that missing link. So here's a map of our samples. We had 3 sample stations, one proximal station, which was very close to the wellhead, one distal station, which was a bit further away, and then one uncontaminated station, which had no oil in it. So as I mentioned, these were all water samples. They were pre-filtered, and then the microbial community was collected on a 0.2 micron filter. And then DNA was extracted using the phenylchloroform method, and the DNA and RNA were purified. And so once again, my main workflow steps: obtain MAGs, search for relatives, and do read mapping. So here is a table of all of the microbes we used in this analysis. I definitely don't expect you to read this, but some key points to take away. The names in bold are the mags that we assembled. The names not in bold are their close relatives that we found through whole genome phylogeny. These first three groups were already known to be present and abundant during the Deepwater Horizon spill. So at the top, we have Bermanella, which was previously ocean spirales, and then Cycloclasticus in the blue and Colwellia in the purple. Also, if you could read this table, you would see that these microbes were found across the global ocean. So of course, our mags were from the Deepwater Horizon spill in the Gulf of Mexico. Some others were assembled from samples using Deepwater Horizon mesocosms. But there are also some from the Red Sea, Arctic sea ice, Mediterranean Sea, South Pacific, South Atlantic, and some crustal fluids as well. So it's important, once again, to remember that all of the microbes in this table were compiled just by looking at close phylogenetic relationships. But after we do gene annotation, we can see that all of them encode for hydrocarbon degradation pathways. And so here is a map showing hydrocarbon degradation pathway in the shape and the taxonomy in the color. So if we focus in on our prime area of interest, which is the Gulf of Mexico, you can see that Bermanella in pink encode for medium chain and long chain cyclic alkane degradation. Cycloclasticus and Colwellia encode for all hydrocarbon degradation pathways, while SAR encodes only for aromatic and UBA for long chain cyclic and aromatic. And so when we take a step back and look at the map as a whole, we can see some clade substrate specific utilization. So for example, all Cycloclasticus encode for gaseous alkanes. So that's why you see a lot of the light blue squares. Additionally, all of the SAR mags only encode for aromatic hydrocarbon degradation. So now we're going to take a closer look at some of these pathways. So this is one of the main heat maps from this paper. At the top, you'll see the log heat map key and then the taxonomy color code key. Below that is a color bar denoting what type of hydrocarbon is in that section. The genome names are below that. The ones that are colored represent the mags that we assembled, while the black text represents their relatives. The taxonomy color bar is underneath that, and then you have the heat map. So here, it starts with the metagenomes or the DNA. So remember that tells you about abundance. And then underneath that is the metatranscriptomes or the RNA, and that tells you about activity. And then the last color bar is represents the different genes of interest. And then below is another heat map with different hydrocarbon groups. Okay. Also for the heat map scale, the darker colors mean lower values, while the lighter colors mean the higher values. So starting with gaseous alkanes, over here, we were specifically looking at methane. Methane was one of the most abundant hydrocarbons released at the beginning of the spill. And so we're looking at methane monooxygenase. And as you can see in this heat map, you'll see a lot of blue highlighted in that red square. All of the Cycloclasticus genomes in this study encoded for all parts of the methane monooxygenase enzyme. And so this is important because it's a new metabolic pathway for this group, but also this group had recently been reclassified into the group containing known methane oxidizers. So this supports their reclassification into that group. Next, we'll move over to medium chain alkanes, which in this case, we are looking at C8 to C16. And for those, the main enzyme we're looking for are alkane hydroxylases. So there's two main ones. The most well-known is alkane monooxygenase, or ALK-B, but there's another one called cytochrome P450 alkane hydroxylase, or CYP153. And so if you look in this part of our medium chain alkane heat map, you can see the pink that corresponds to Bermanella. And you can see that they are very abundant and active in this pathway. What's interesting, though, is that the Bermanella that we assembled encoded for the CYP153 instead of the more common alkane monooxygenase. And this gene has actually been shown to be upregulated in other environments with hydrocarbon contamination of medium chain alkanes. And as I mentioned before, these were some of the most abundant hydrocarbon types in the Deepwater Horizon oil spill. Next, we'll go to aromatics. Aromatic hydrocarbons are the ring structures. And what we found here is that microbes that were not previously known to respond to the Deepwater Horizon oil spill did in fact have the potential to degrade aromatic hydrocarbons. So that SAR 324 group and the UBA group. We're going to take a quick pivot. I bet you didn't think you'd learn about beer today. But during my PhD, I had a really great advisor, Olivia Mason. And as I mentioned, all of my PhD research was computational. So I was going a little stir crazy, just coding and writing. And so she agreed that I could get a part-time job. And thankfully, I found a local brewery who had a quality lab. And that was how I was able to keep my lab skills sharp during my PhD. And so we tested all steps of the brewing process. We looked at the proportion of milled grains. We tested the pH, the dissolved oxygen, looked at yeast vitality and viability. We also did plating and even qPCR for some of our samples to make sure either there was no yeast or mold or bacteria in the samples, or the right kind of bacteria was in there. We also did one test called diacetyl testing. So fun fact, diacetyl is a natural product from yeast fermentation, but it's actually also the compound used in microwave popcorn to give it the butter flavor. So if there's too much diacetyl in a beer, it can produce some off flavors. So just fun fact for you. So moving on to my work here at Dissel NUSA, I work with doctors Kiel Reese and Jeff Krause. The main project with Brandy is the microbial cycling of methane hydrates in the Ross Sea. And then Brandy and Jeff are co-PIs on the project involving microbial effects of reverse weathering. I feel like I've done a lot in my year and a half here so far, but one of the very first things I did was become certified to handle radioactive material, because we do use radioactive silica 32 in a lot of our experiments for this project. So a quick introduction to the silica cycle. Silica, or silicon, is the second most abundant element in Earth's crust and the seventh most abundant in the universe. And if you've ever talked with Jeff about silica, one of the first things he'll say to you with great enthusiasm is diatoms. So diatoms are phytoplankton. They provide a lot of the oxygen we breathe. They form the base of polar food webs, and they require silica in order to build their frustules. And so because these phytoplankton require silica, that means the silica cycle also has a lot of interactions with the nitrogen, phosphorus, and carbon cycles as well. So here's a schematic of the silica cycle in the marine perspective. So here you'll see the white arrows represent silica sources. So we have rivers, submarine groundwater, upwelling, and glacier meltwater. Silica sinks are in orange, and you'll see we have reverse weathering and sponges. Biological fluxes are shown in green, and for those you can think of the diatoms, taking up silica and then being exported to the bottom of the ocean. And so our project revolves around reverse weathering. So when you hear reverse weathering, you might think weathering of normal rocks, which is when you take a big piece and break it up into smaller pieces. And reverse weathering is in fact the opposite, where you take smaller pieces and form in situ silicate clays. So here is a schematic of what that looks like. It takes silica, aluminous clays, iron oxide, organic carbon, soluble cations, and bicarbonate, and you form new clay material along with carbon dioxide and water. And so some of you might notice right away that we're consuming alkalinity and we're putting more CO2 in the ocean. So this could have implications for a changing climate. Reverse weathering also traps silica, which prevents it from being available to diatoms to fuel the biological pump. And it's overall a poorly understood process, so we're not really sure how it affects cycles of other elements, including lithium, aluminum, potassium, and iron. So this brings me to some field work we've done so far. So we've had 2 cruises in the Gulf of Mexico so far. One in September 2024, which was 3 to 10 days after Hurricane Francine. And then January 2025, which was 5 months after Hurricane Francine. And we're hoping to have one more next year. For these projects, the radioisotope experiment was handed off to other members of JEP's lab, while I worked with Penny Dimitriadis to do microbial sample collection. Earlier this year, I had the honor of going to Antarctica. So the main purpose of this cruise was for the more microbial sample collection for the methane hydrate project. However, we all have side quests, and so I did a silica experiment while I was there as well. So here you can just see a time lapse of us going through the ice before it got too thick. The next column shows some collaborators enjoying a beautiful sunset. There's Lydia, Kristen, and I wearing our float suits. Lydia taking oxygen from the sediment core, and then our science party photos at the bottom. And of course, I had to include a selfie because I'm a millennial. So the expedition I'm going to spend a little bit more time talking to you about is the Arctic expedition. So I went up there in July of 2024. You can see the map on the bottom has the pin where Svalbard is. The top left photo shows a little research town, and I do mean little. It takes about 5 minutes to walk through it. Part of being in Svalbard is that everyone is required to do a polar bear safety and weapons training course. So the next picture is my group leaving the shooting range. That was indeed my first time ever shooting a gun, and the fact that it happened in the Arctic Circle, I think, is pretty awesome. And then some other pictures. The middle top one is of a collaborator doing some sampling in what he called a river, I would call a creek, maybe. A couple other cool things about when I was there, it was midnight sun, so there was sunshine 24/7, which was really weird when I was leaving the lab at 2 a.m. And I did get to see a polar bear, so that was another highlight of the trip. So now we're familiar with where our system is. So how does silica get from the glaciers to the fjord and coastal ocean? So this is done by glacier weathering. So that transports the biologically available silica from the glaciers to the fjords and offshore environments. However, geochemical processes can sequester this dissolved silica, prevent it from getting to the fjord and coastal ocean, and then fueling diatom growth and the biological pump. So in terms of sampling, we did small CTDs, as you can see in the upper left-hand corner. We did sediment grabs shown in the upper middle photo. We also deployed some Niskin bottles in the bottom left photo for our water sample collection. And we did get a couple cores as shown in the bottom middle photo. And then those were all done on the MS Tyston, which is their workboat. It was large enough to fit 4 scientists, so very nice and cozy. And then the rest of our collection for the Glacier River samples, for example, we used their small boat called the Polar Circle that we could attach a small winch to if necessary on the side. And so the 2 photos on the right are from the Polar Circle. And then just to orient you to our naming scheme, the KB samples you see in our sample map represent forage samples, while the GL ones represent glacial rivers. So here's our experimental setup. You can see in the table on the left, we have 2 main categories, live and dead. At the top of the live, you'll see 10 SSR. That stands for our 10 grams per liter for our solid solution ratio. We also, of course, included blanks, which was just regular seawater. For our dead treatment, we had the same 10 grams per liter solid solution ratio, but we added ZN, which here stands for zinc chloride, which is a biocide, which would kill all the microbes, as well as heaps, which was a buffer. And then we also had blanks for that treatment, which was the zinc and heaps with the seawater. And so this video on the right is just me making a stock solution of most likely the dead 10 SSR treatment and dispensing it into our tubes. So for each time point, I would take a small bit of our tube and filter it. We would collect the particles on the filter, and that will be used to look at the radioactivity in those particles using what we call a beta counter or a multi-counter. We also collect the filtrate, as you can see here in this part of the video. And that will be used to determine dissolved silica concentrations, as well as the radioactivity in the solution. And so the photos on the top, you can see I would take the filter off. I would put it on a plastic ring called a planchette, let it dry, and then wrap it in these different types of foil to be put into our beta counter. So here's just a different look at what some of our sediments will look like. You'll notice the different colors and grain size associated here. Once again, KB stands for fjord sites, GL stands for river sites. And just to highlight some of the differences in our sites, you can see that the GL3 site, the photo up top, the glacier is a marine terminating glacier, which means it comes right up to the marine environment. While GL1, which has more of a red coppery color, comes from the Red River. And you can see that it's a land terminating glacier. So that glacier was way far away from the marine system. All right, so to get into some results, first we're going to look at dissolved silica in the river sediments. So on the y-axis, we have average dissolved silica in micromolar. And on the x-axis, we have our day or time point. So all 3 of these plots are on the same scale. You will notice that GL2 and GL3 only ran for 4 days, not 6, just due to time constraints. So here our alive samples are green and our dead samples are orange. The open shapes represent our blanks or controls, while the colored in shapes represent our experimental. And so one basic takeaway you can see here is that the orange line is at top. So there's more dissolved silica available in the dead treatments. So when there are not microbes available, or when there are no microbes, there is more dissolved silica available. This is showing the same information just for our fjord sites. Take a look at the y-axis. You can see it's much bigger than our previous one. But these data all tend to follow the same trend where there is more dissolved silica in the dead than the live. And so we can put that all together so you can see. Remember, keep in mind of the different y-axes for rivers versus fjords. And what you'll notice is that we seem to have this one outlier here that's a little bit different. This was our deepest fjord site and most marine influence fjord site. So there's a lot to dive into here, but that could have some, there could be some interactions there as to why this one didn't follow the same general trend. But overall, we can see that the net release of dissolved silica was an order of magnitude higher in the fjord samples compared to the river ones. Next, we'll look at some radioactivity in the filtrate. So here on the y-axis, we're plotting the DPM value, or disintegrations per minute. And so DPM is a unit of radioactivity that measures the actual number of radioactive atoms in a source that decay in 1 minute. And so here we're looking for negative slopes because we're looking to see consumption of the isotope. The river samples seem to look a little bit different from the fjord samples. In the fjord samples, we see much faster consumption. You can see that the slopes of those lines are much more extreme compared to the ones in the river samples. And so what does this tell us? This tells us that radioactive silica consumption was mirrored in the trends shown in the dissolved silica data for the fjords, but not the rivers. So that will be something we'll be looking into more. And so what's the main point here? So we're seeing accumulation of dissolved silica and removal of the radioactive silica from these samples, which shows us a two-way exchange of sediment silica uptake and sediment silica dissolution. And we'll still have more to look into that to see whether it's precipitation and sequestration or absorption and release. And the overall big picture here is that the spring diatom bloom in the Arctic is one of the biggest PCO2 drawdown events. The dissolved silica can be delivered to the fjord, and it's affected by a lot of different processes, including glacial meltwater, riverine discharge, and geochemical processes. So any alterations in the dissolved silica delivery to the fjord and coastal ocean can have consequences for primary production, phytoplankton diversity, and ecosystem structure. So what's next, besides more data analyses, of course? We are going to be looking at the biological role in the silica cycle for reverse weathering. So here is a schematic from our proposal that shows the conceptual model of microbial effects on diatom silica dissolution. So we think that microbes might play a role in this reprecipitation of silica, maybe by organic matter degradation, changing of sediment pH and redox conditions, or just competing with abiotic reactions for elements such as iron. So here is our schematic again that I have showed you. And this work we'll be doing will involve all parts of this time. So something brand new for me will be culturing bacteria. I started some cultures last year that I definitely need to check in on. I'm looking for an intern to help me with this part of the project, where we'll do mock community sediment slurry experiments. So we'll put in 2 known bacteria separately and together and see what their interactions are in terms of this reverse weathering. And then, of course, we're doing our culture independent methods with all the samples we've collected in the Gulf of Mexico, as well as the Ross Sea. And then once we have all the sequences, back to my normal workflow, obtain mags, search for related genomes, and determine abundance and activity. One of the things I really love about being here at DISL is all the mentoring and outreach opportunities I've had. I've done more in a year and a half here than I did in all of my PhD. So I've actually been able to hire my own interns. So my first hire was Teravia, who did such a great job. She is now the lab manager of the Kiel Reese lab. I worked with my first NSF REU student, Cole, this summer. And then my very first undergraduate intern was Natalia, and she was so impressive that we invited her to come out on a cruise with us. I've also participated in Discovery Day. I wasn't able to this year because I was in Antarctica. Jeff has formed a strong collaboration with a Missouri class in Missouri that we've worked with twice already now. Brandy and Lydia Hayes Gustella set up a ship to shore while we were on the Antarctic cruise for a local elementary school here in Mobile. And then just this morning, I actually did an outreach event to a second grade class in Florida, telling them about my Antarctic research, mostly just showing them pictures of animals we saw. And so my scientific path has really helped me fulfill my ultimate bucket list of traveling to all the continents. And then I made this schematic the other day just to kind of show you my path. So I started out, of course, with the charismatic megafauna, studying sharks. Then I went to this air-sea interface. I've made my way through the water column, and now I'm at the sediment water interface and looking at the subsurface. I do have some publications out there. My first three have some double-digit citations, which I'm very happy about. I'm still working on getting the last chapter of my dissertation published, but hopefully it will at least be submitted next week. And all of this is to say I could not have done any of this work alone. So quick thank you, of course, to Brandy and Jeff and their lab members. It's not easy learning something new or doing something you haven't done in a while. I haven't done DNA extractions since 2016, and so I'm learning how to do them all over again. I want to, of course, thank everyone in Svalbard for their collaboration, especially Dr. Kate Hendry, the captains and crews of the ships I've been on in the last year and a half, as well as the Gulf of Mexico Research Initiative, which funded my master's and PhD, and the National Science Foundation, which funded my expedition to Africa and the projects I'm working on now. With that, I'll say thank you. And one more quick plug. My contract here is up at the end of 2026. So now's the time for me to start looking for my next position. So if anyone wants to have conversations about that or ideas, please feel free to come up and talk to me at any time. Thank you.